So, it is a big pleasure for us to host today in our Bar-Ilan University Vision Science Seminar Professor Chris Baker from NIH. Professor Baker received his BA in Neuroscience from the University of Cambridge in England and then continued there to an MA in Neuroscience. He has a PhD in psychology from the University of St. Andrews, where he worked with Professor David Perrett on neurophysiological studies of visual perception. He did a postdoc at Carnegie Mellon, where he worked with both Professors Carl Olson and Marlene Berman on combined monkey neurophysiology and human behavioral studies of visual object representation and learning. He continued to another postdoc at MIT, working with Professor Nancy Kanwisher using functional brain imaging techniques to investigate learning, plasticity, and high-level vision in the human cortex. After that, he joined NIH, where he's now a senior investigator. He is the chief of the unit on learning and plasticity in the Laboratory of Brain and Cognition, where he aims to better understand how the structure, function, and selectivity of the cortex change with experience or impairment, even in adulthood. He investigates the nature of perceptual representation in the human brain, focusing on complex visual stimuli as faces, bodies, scenes, and words, how experience and learning change the neural and cognitive representations of sensory input, and how the cortex adapts following damage to the nervous system, either peripheral or central. His lab uses a combination of functional imaging techniques as fMRI, MEG, TMS, as well as behavioral methods and eye tracking to achieve these goals. He publishes in top leading journals in the field as Nature Neuroscience, Nature Human Behavior, Nature Communications, PNAS, Memory, Trends in Cognitive Sciences, and more. He has more than 22,000 citations and an H-index of 68 according to Google Scholar. He has mentored dozens of postdocs, graduate and undergraduate students, as well as summer and high school interns. Many of his postdocs have transitioned to tenure track positions in multiple distinguished universities in Europe and in the US. He has received multiple mentoring awards. So without further ado, although there's so much more to say, we're pleased to have you today with us, Chris, to hear about your exciting research, and thanks so much for joining us.
Okay, well, thank you for that very kind introduction, and I'm very happy to be here. And this has been a great vision seminar series, and I'm happy now to be part of it. So today, what I'm going to talk about, as you can see on my title slide, is object vision in a dynamic world. And to give you an idea of the perspective that I have, I mean, the fundamentally my interest is saying, how do we take complex visual information that's out there in the world and understand things in a way that allows us to perform adaptive behavior, whatever we're trying to do in that situation. And there are many different things that I could have talked about today, and I thought about it hard. And actually, what I'm going to present about today is all new work. None of this is published. It's all very different things. So what you're seeing here is a little bit fresh. Some of it may not be fully digested, so please kind of bear with me, and I'm happy to take any questions as I go along to clarify what I'm talking about. So from a vision perspective, we can think about a complex visual scene like this, and we'll say, okay, well, how are we going to understand how we represent this scene, how the brain is taking the information and producing a representation that's useful from a behavioral perspective? And so, what we often like to do is to say, okay, let's take very highly controlled stimuli. So we can say, okay, let's do a vision experiment, and we're going to present nice, tightly controlled visual stimuli. And we can look at this and we can say, okay, well, this is great. We've got these nice controlled experiments, but actually we're missing lots of the features that are present in the real world. So we'll shift to more complex visual stimuli. So for example, here, some stimuli that Jack Gallen used, I guess 30 years ago now. Or we can go to slightly more complex stimuli that are more like objects. These are ones that I used while I was in Pittsburgh. But these aren't like the things that we see in the real world. So we can go to like, okay, these are objects. These are nice, tightly controlled stimuli of objects. But again, this misses lots of the features of the real world. So we're going to shift to photographs. And so we can do experiments with these kind of visual stimuli. But then we may also want to recognize that, in fact, objects don't appear in isolation like this. They don't have these nicely cropped outlines. They actually appear in visual scenes. And so there's a tendency in vision science to sort of go and try and say, okay, we want to sort of balance like our control for the experiments we want to do, but we also want to recognize the nature of the natural visual world. And so whatever types of stimuli that we present, we may do an experiment like this. So we're going to bring participants into the scanner, into the testing room or wherever. We're going to put a cross on the screen and we'll say, okay, please fixate. and then we're going to do this. So now something rapidly flashed on the screen. Now you have no context for what I was about to show you, but yes, you could understand. And probably, I'm not sure what you took away from it. You probably saw there were a bunch of birds, there was grass, there was something going on there. Or we could do something else, and here's something else, right? And you can fully understand what is going on here. And I think in terms of thinking about sort of high-level object vision, these are sort of like, this is the classic style of experimentation. And so what we're talking about here, this kind of classic experiment, is we're going to have people fixating because we want to understand where information is landing on the retina. We're going to use these sudden onset stimuli, and these typically are often static stimuli, particularly when we're thinking about visual object recognition. But all of these kind of characteristics are things that maybe we should consider because maybe they're problematic for thinking about natural vision. So in terms of fixation, well, our eyes are rarely still. We're constantly moving our eyes around. Fixation is a very unnatural situation. Sudden onset stimuli. Well, this doesn't actually really occur in natural viewing. And I don't think that sometimes people argue that when we make saccades, it's kind of like we have this sudden onset of stimuli at the fovea, but I really don't think that's not quite analogous. So we don't really have these sudden onset stimuli. These rarely occur, and things basically aren't static. The real world doesn't just sit there. Things are constantly moving. And so these are kind of things that, throughout my career, I mean, I've done these kind of classic object vision experiments. And more recently, I've been thinking like, well, okay, we've done these experiments, but what does it actually mean for trying to generalize to natural vision? Now, of course, the trends in the field is to go towards, okay, lots of people recognize this. And so now we'll move to a situation where we say, okay, let's do naturalistic movie watching. So now we'll show participants segments from movies. And so these, people like these are naturalistic. They have there's lots of dynamic information. These are very complex visual stimuli. They may actually be presented where there's both audio and visual material. They have narrative structure. We can define events in them. We can do lots of different things within these. And we can also have people free view. So this ends up being a much more complex kind of situation. Although I would argue that actually it is worth questioning how naturalistic an edited movie is. And I think editors of movies are actually amateur psychologists because they have to understand how you direct people's attention around the screen. But with movies, while they're so complex, it's difficult now to understand, well, okay, well, how do we understand what we've learned from sort of static visual processing, and how does that generalize now to the movie watching? Is this just a completely different situation where it's very difficult for us to try and relate these things? And I think one of the critical questions that we have to ask is how much of what we have learned from classic object vision studies actually generalizes to natural viewing. And that's kind of one of the things I want to really address today and sort of say about how we've been thinking about this problem and trying to address it. So to give you an idea about why we might be concerned about this, what I want to just present quickly is a study from David Leopold's lab from a few years ago, from 2023. So this is actually work in non-human primates, and they're doing electrophysiology. So they showed movies to monkeys, and these were movies of sort of natural history movies of monkeys moving in the environment. And they recorded neurons from two face-selective patches in monkeys. It doesn't really matter what these patches are, but these are patches in sort of anterior temporal cortex in monkeys. So they've got the neural data over trials as the monkeys are watching these movies. And what they did is they presented the movies either as the original movie, showing up here, or they could present individual snippets from the movie that might now be presented out of order. They can get the neural response to the individual snippets, and then what they did is they reordered those and tried to see, can they reconstruct the neural response to the intact movie from the response to the individual snippets? So here what they're showing is this is now the firing rate. The black line is actually the response to the movie, and this red line is their attempt to reconstruct the response from the response to the snippets. And in this case, they're taking the early response period to those individual snippets, and you can see it's not a particularly good fit. If they take the later period from those snippets, they do slightly better, but it's still not a great fit. And I think particularly interesting is now if you go to the static frames, and they just took the central static frame for each of those kind of snippets, you actually do a very poor job of predicting the kind of the response that you see to the movies. And in this paper, what they say in this paper is they suggest that the results suggest that a model of visual function based on discrete and randomized visual presentations may not translate well to the brain's natural modes of operation. If this is true, I think this is actually really important for us to understand so we know how much of what we have learned about vision actually is relevant for natural viewing. Now, one of the challenges with this study, I think, is that because they were just sort of chopping up and changing this movie here, it's actually hard to know exactly what accounts for that difference. Why can we not do this kind of prediction from, say, the static or from the snippets to the full movie? And what they focus on here is actual temporal continuity. So they argue that actually there's a major role for temporal continuity in sort of processing in natural vision. Oops. So in terms of thinking about observation in a dynamic world, what I'm going to do is present two different sort of angles on this. And the first angle is going to be say, okay, let's think about what it means that the world moves, right? So we'll focus on that and try and see what can we say going from static to dynamic when we think about the world move. And then in the second part of my talk, I'm going to say, okay, what about when we move? So sort of break it down to these two different kind of things that we might want to consider. So when we started to think about sort of dynamic visual stimuli, I'd always been very hesitant to go to dynamic visual stimuli. And if I'm honest, the reason is because it gets complicated. Right? Now you've got something else that you want to try and control. And so when we started doing this, we actually sort of focused on areas where sort of dynamic is actually really important for what we're actually looking at. And so the first thing we focused on was looking at facial processing. And what we were really interested in understanding, and this is work that was led by Rohini Kumar, who was a postbac student in the lab, and Trudy Chepi, who is a staff scientist in my lab. And what we wanted to do is to try and understand the temporal dynamics of identity and expression processing. And the way we were thinking about this is to say, okay, identity is a fixed property of a person. We can have a video of a person doing something, identity is fixed, but expression is a changeable feature. And really, expressions, although we can have static expressions, an expression is a dynamic property. So we felt it's very important to sort of move to sort of dynamic kind of visual stimuli. And so what we did in this study is we had 6 different identities, and these are the 6 different faces that we used. And we had 6 different expressions or facial expressions that these faces could be making. And so you can see here, happy, surprised, angry, fearful, disgusted, and sad. And so we can combine the identity with the expression, and we created these movies. These are just 1-second movies where the face will go from a neutral to one of those 6 expressions. Now, in the study, people are looking at these kind of expressions, and we have them do a task where actually what they are doing is they are telling us anytime they see what is an unnatural expression. So this is an expression that has facial movement, but it's not like a natural facial movement. So it'd be something like this. Now, because we're particularly interested in dynamics, we wanted to use a method that actually will give us that kind of high temporal resolution. So to do this study, we turned to MEG. And just to give you an example of how the experiment goes, this is from the subject's perspective, and I don't know how. I hope these videos aren't too choppy across the internet, but this gives an idea of what people are participating. And every time they see one of those unnatural expressions, they're just going to tell us that they see one of those. So using MEG, we can collect high temporal resolution information about the processing in the brain. And what we're getting is actually for each of the sensors, we have roughly 270 sensors in our MEG system. We get this dynamic readout of kind of the response to the individual trials of these facial expressions. Then what we're going to do is we're going to take the pattern of response across all of the sensors. So we're doing a whole head analysis here. We take the pattern of response across the sensors. We'll use a chunk of those trials for training and a chunk for testing of a linear classifier. In this case, say this is a classifier where I'm just sort of demonstrating as if we had 2 sensors, we have 2 identities. We're going to build some sort of linear classifier, and we're going to repeat this at every single time point. So we're going to get this dynamic readout, and we're going to repeat this for every single participant. So we can now get kind of a readout of our ability to decode identity and expression information over time. So here we go for identity. So the stimulus, the videos were 1 second long, shown by this here. And we're just looking now at ability, our decoding accuracy. Because there are 6 identities, our chance level is 1/6, which is shown here. And this is data averaged across 24 participants. So we can produce a curve that looks like this. And this kind of actually is sort of familiar, maybe familiar to people from looking at other MEG experiments. Where what we see, we have some sort of baseline. We can't really do any decoding early on. Then we see this rapid rise to an early peak. There's roughly around 154 milliseconds, and this kind of slowly decays over time, over the course of the stimulus presentation. So this is what we see for identity. Another way we can look at this and say, okay, this is now what I'm doing here is we're training on a given time point and testing on the same time point. But we can actually train and test on different time points. And this allows us to look at something that people tend to refer to as temporal generalization, and allows us to say how stable is the representation over time. So we can produce a plot like this. And so the way to think about this, we have training time on the y-axis here, we have testing time on the x-axis, and this plot here actually corresponds to this diagonal over here. Points off the diagonal, for example, out here, this is where we're training, say, on 250 milliseconds and testing on 500 milliseconds. And so here you can see we can actually still do this, which suggests that whatever we're learning at 250 milliseconds generalizes to some extent to 500. But by and large, what you're seeing here is this strong diagonal. So this is what we see for identity. So now what about expression? So now expression, one thing to note here is expression isn't present at the start of the movies. The movies started with neutral, and so this is actually a dynamic property of the movies. And maybe not so surprisingly, what we see here now is it takes long before we start to see any signal. Around about 250 milliseconds, we start to see our ability to decode which of the 6 expressions was present. And this is sort of peaking around 668 milliseconds, but actually is quite stable for this long period of time. So we have this different kind of profile for identity and expression. And now you might wonder, well, okay, because the expressions are developing over time, are we seeing this kind of prolonged kind of process? Because actually maybe the information isn't present in the stimulus for quite a while. So we did behavioral experiments to say when during these videos can people first see the expression. And that's shown here. This is kind of like estimates the times for the different expressions where people are first able to tell you what the expression is. And so actually this still occurs quite early on. And so we get this prolonged kind of period where this is staying. We get this elevation. If we look at now the temporal generalization, what you'll see here is it's less diagonally than the identity. So here we actually see much more generalization, particularly in these later time periods of the information that's present in the response. So here we have these two things. This is kind of interesting to us, so we can look at identity and expression, but one of the things we don't know is, well, okay, is this profile for identity? How much of that is really reflecting how identity is processed, and how much of that just reflects the fact that identity was present at the sudden onset? We've gone from no stimulus to having a face that has an identity. Expression is slowly developing. Is this telling us something about how we process identity and expression, or is it telling us something more about the nature of the visual stimulus and how that's changing over time that may not necessarily reflect these two things? So it's kind of hard to address that in these kind of data. And so what we saw here was, if you think about temporal dynamics of identity expression processing, for identity, we see this rapid onset, we see weak temporal generalization. For expression, we see this slow emergence that actually is consistent with the fact that it's actually slowly emerging within the stimuli. And we get this sustained temporal generalization. But we want to sort of understand a little bit more of what's happening and how the nature of the stimuli affects what's going on. And so we wanted to go to a slightly different type of visual stimulus. So here, on the sort of points towards, say, the full expression, it's hard to know exactly what do those midpoints really mean. What does a midpoint of, say, a fear expression actually mean? What is actually in the stimulus? We wanted a bit more control over that. So here we moved away from kind of facial processing to look at something where actually we can say what is in the stimulus at any given time. And so here we've started to look at head orientation. And one of the reasons we wanted to do head orientation is because we already knew that we have information about head orientation in the MEG signal. And this is work that was actually led by Sebastian Montesinos, who was a postbac student in the lab, and he's now just started a PhD at Princeton. So here we can create tightly controlled static stimuli, tightly controlled dynamic stimuli. So these are examples of static frames of our stimuli. These are artificially generated heads that gives us very precise control now that are occurring. And we can present these at kind of different orientations. But we can also take these different orientations and make a movie. So now we have this head rotating smoothly about this. And this head here, as it rotates, is passing through those orientations that I'm showing in the static in this particular case. So these are the kind of stimuli that we're working with. And the key question is, can we generalize from whatever we see for the static stimuli? Can we say train a classifier and the responses to the static stimuli? And can we now read out orientation during this dynamic movie? And when can we actually do that? So we had 16 different static orientations that went all the way around. So we go from the front of the head to the back of the head. So 360 degrees sort of rotation in depth. And for the dynamic movies, 8 of these orientations could be the starting point for a movie, and every movie went through 180 degrees. Okay, not a full 360, but 180 degrees. And so we're going through this set of orientations, and we can go through, for the dynamic, we're obviously generating the in-between orientations as well, and we can rotate in either direction. We can go rotating in the positive or negative direction either way. For a given trial, so we start with one orientation, and then it's going to pass through 8 others until this 9th orientation at the end here. Each individual trial lasted 3 seconds. So 3 seconds of rotation now. So first of all, what I want to say is, okay, let's just look at the static. Let's say, okay, let's look at the static processing, and actually let's break down what's happening over time during static and try and understand that static response before we think about how that might generalize now to the dynamic situation. So we can do what I showed you before with the identity and expression data. So now what we're going to try and do is we're going to try and decode what is the orientation of the head. And we're looking at this over time. So in this particular case, the static orientations were present on the screen for 300 milliseconds. So here we get some sort of decoding curve that we can see here. And again, this looks like what we sort of expect. We're at baseline down here, the stimulus comes on. We actually start sort of seeing some evidence for decoding of orientation around about 90 milliseconds. And then this is peaking around about 124 milliseconds. And then there's also this sort of dip and this slow rise again. And actually, this is very common, I think, what we see when we're looking at kind of MEG data and looking at this kind of decoding. But we have this kind of second phase of the processing, the second peak here. The peak here actually occurs at 329 milliseconds. So when we look at the static, we can actually say, okay, we've got this kind of early phase of processing. We get this early peak, and we get this second phase of processing. We get this later peak. And what I want to say is, well, what's actually different about kind of what's happening in these two situations? So we can also look at like our accuracy as a function of the actual orientation that's present in the static head. So here I'm going to show this kind of rows plot, and we're going to show for each of the orientations what is the accuracy. If we now focus on that early peak, we'll get an accuracy that kind of looks like this here. And what you'll see is like, I mean, we do pretty well for all the orientations. We do a little bit less well for the back of the head. You get the strongest kind of decoding for these left and right orientations, 90 and 270, and you also do very well for the front-facing orientation. So it's a little bit anisotropic, kind of like the processing that you get here. If we now look at what happens for the late peak, okay, we do less well, right? Decoding is a little bit worse, but interestingly, it's also a little bit more isotropic. We actually get more similar representation around all those kind of orientations. So you can see the drop-off from the early to late is larger around here than it is for actually around towards the back of the head. So this late peak shows weaker, but more isotropic representation. We can also look at this in terms of like that temporal generalization I showed you before. So you remember here, this is now we're varying the training time and the testing time. And this diagonal through here would correspond to the decoding profile that I showed you before. And you can see here, this actually looks kind of similar to what I showed you for the identity processing before. We actually see this sort of early peak around here, and there's a later phase where there's a little bit more generalization around here. To sort of make this clear, what I can do is take slices through this plot. So this is sort of corresponding to where we see that early peak. If I now draw through that one training time across testing time, you'll see a profile that looks like this. So here's this sort of sharp peak. We get some limited generalization around this peak here, and we get a little bit of generalization to a later point in time, which you can see out here. If we now look at the second peak, and I draw the same profile through here, you're going to see this green line kind of here. What you'll see here is around that time of that training, we actually get sort of this broader generalization than you see for around the training time here, but you also generalizes a little bit to earlier on here. And in general, we're doing better out here than we are from the generalization from that early peak. So this late peak shows a tendency for this greater temporal generalization. So that's just sort of saying, like, what do we see when we're looking at the static processing and trying to see what's happening there over time? But the real question is now, okay, what about going from the static to the dynamic? And there's two kind of important questions here. So the first question is going to be, well, can we actually, if we do the static decoding, if we train our classifiers on the static stimuli, can we do anything in those dynamic videos? And I think when we started this, we're not sure what the answer to that is. The second question is, okay, if we can do that generalization, right? And the study that I showed from David Leopold may give us cause for concern here. If we can do it, which part of that static response is showing greater generalization? And, one way of thinking about it is that actually that second part of the response may reflect kind of like the more stable part of the response that may then show greater generalization later on. It may be that that more sustained kind of processing is more relevant for natural viewing. So this is what we wanted to kind of look at. So here, what I'm going to do is I'm going to take, this is just showing what the decoding was like for the static stimuli. And we're going to take a sliding window approach. We're going to slide a window through time and then look at, as a function of training time, what's our decoding accuracy within the dynamic videos for the orientation. And we're just looking at the specific, we take the orientations when they actually occur. We're assuming that's the correct response at that time. We'll say, what is our decoding accuracy for that? And we'll actually get a curve like this. So the first thing you can say is, okay, we can do this to some extent, right? So it's not the case that nothing from static is generalizing to dynamic. This is kind of important. It's kind of reassuring that there's something going on there. But you may also notice that the accuracy actually here is lower than what I was showing for static to static, and we'll talk a little bit more about that in a second. The other thing is you can see here that while we have this peak that happens around about 130 milliseconds, the generalization is actually stronger for this later part of the response. So training on the later part of the static response, even though that's a weaker part of the response, gives us more generalization into the dynamic videos. So does static decoding generalize to dynamic videos? So the answer here is yes, but it's weakly compared to within static decoding. And which part of that response shows greater generalization? It's the later part of the response. So to get into this a little bit more, we can think about, okay, let's think about in a given dynamic video. What we're actually doing is we're going from one orientation all the way through. These are the orientations to one later on, right? And these are the orientations that we trained our static decoder on. And now we can say, okay, well, let's have a look at how our decoding looks for each of these kind of successive orientations. So like the serial kind of processing of the information. So let's say, okay, we're going to look at like our ability to decode the first orientation in a video. Now in the video, this is actually the onset orientation. So in a way, this is the most similar to just a static presentation. The difference is that this is the onset orientation that then immediately starts moving, whereas in the static, it would stay still. So if we look now at our ability to decode that particular orientation, and to look at that, we actually see this very nice sharp peak here. It's happening around about 130 milliseconds and dies off pretty quickly. So this says, okay, that static, the decoding from the static to the first orientation in the video is pretty good. Well, what about if we now look at the second orientation? Well, actually, this is a lot worse. So actually, our ability to go from the static to that second orientation is worse than what we're seeing for what I would argue is more like the static to static. This is for the second. If we go to the third, and we can go and look at all of them together there. So what you see is that generally, what we learned from the static generalizes much better to the first frame of the dynamic video than to anything else. But there is stuff going on here, right? It's not that there's nothing here. And what we're going to do is I'm going to get rid of this big peak here so we can actually look a little bit more closely at what's going on. So if we now say, okay, we're going to focus just on that early part of the static response, and that's what I've shown you in the last slide. This is now looking across the order of the presentations, going from, this is now the second orientation, the third, and then coming all the way down to the ninth. And you'll see that for this yellow one, which is the ninth one, this is the final orientation in the movie, and this ends sort of about 200 milliseconds after the movie ends. One thing you'll notice here is that as you go from the second to the third all the way through, we tend to see this reduction in our ability to decode. So our ability to decode is decreasing as the movie goes on. Well, what about for the late part of the static response? If we now train on the late part of the static response, this is what we get here. And so you'll see in general, we actually do a little bit better, particularly for those early kind of orientations that appear in the video. And this trend where you go from the second to the third to the fourth, you can see this a bit more clearly here. So we're reducing as we go through time. So again, here what you can see is, yes, we can generalize somewhat from static to dynamic, but our ability to do so is kind of limited relative to what we see for the first frame of that video, and it's decreasing over the course of the dynamic videos. Now, what I've shown you here was kind of like using a decoding approach where we've got these individual orientations that we've kind of been training on, and then our readout is to say, okay, was that orientation, are we correctly estimating that orientation? There's another way in which we can do this analysis that allows us to, for every point in time, estimate what is the orientation, and we can do that across the whole 360-degree space by using a regression approach rather than the approach that I showed you before, which is kind of the classic decoding approach. What this enables us to do is to say at each point in time, we can say, what do we think the orientation that's present based on the MEG response at that time? So this will give us a plot like this. So I'm going to try and explain this plot. So what we're showing here is time is actually going on the y-axis here. So this is sort of like before the movie starts here, and this goes on all the way through to 3 seconds. What I'm plotting now is the difference between the orientation, the angle of the orientation that's on the screen. So 0 would be the angle of the orientation that's present on the screen, and then our estimate of what the orientation is. So negative is going to be it's sort of lagging behind in terms of what we're estimating the orientation is. This way would be kind of like moving forward in time, more like a predictive response. Each row here is kind of looking at across trials. We've got the distribution of the prediction across all the trials. And this white line is showing us sort of where the peak of that distribution is. So what you can see here is that initially as the movie starts, we actually get these kind of nice predictions. They actually are pretty close to what the orientation actually is. And then there's a period here when we're training on the early part of the static response, where it actually seems to be slightly predictive. But as the movie goes on, you can see that we're lagging more and more. So our estimate of what the orientation is falling more and more behind what's actually present in the movie.
Chris, can I
Oh,
ask a question? So
yes.
would zero be the, no, so zero would be like perfect match,
It
like
would be
a
it would be
straight
a
line?
perfect match. And I think one
Okay.
it's so yeah, this would be a straight line. So we've basically reordered everything to put
Yeah.
0 always present at that line so we can look at this kind of lag. And this is why this kind of it can be a little tricky to think about.
Yeah, but just to understand what would the baseline or the truth would be like.
Truth would be vertical here. So
Okay, great.
if you were always able to say exactly what is the orientation at that
Orientation.
point in the movie, it'd be you'd stay down here.
Okay,
So this
great.
is then you're getting further and further away from that. And
Thank
this
you.
is when we're training on the early part of the static response. This is when we're training on the later part of the static response. And what you'll see here is in general, we actually do better. We're training on the later part of the static response. We actually get the distributions are more peaked in general. And actually, you can see that the angle of this is staying closer to vertical than in this particular case here. So the lag is less when we're training on the later part of the static response than training on the early part of the static response. So I hope that's kind of clear. The other thing to remember here, and this is the one thing we're sort of trying to think about how to take this into account, is that we're sort of comparing what is currently present in the stimulus in relation to the MEG response. But we know that there's going to be a latency effect here, right? You would not expect the instantaneous MEG response to reflect what's currently present. We know that it's going to take about 100 milliseconds really to things to really start getting processing in early parts of the visual cortex. So actually, what you might want to do is not compare with 0, but you might assume that actually what you might expect is we would be just a little bit behind, 100 milliseconds behind. And we're trying to work out how to take that into account in terms of the analyses here. But in general, if you were seeing a readout that exactly matched the stimulus, that would already be some sort of predictive response, right? Because of taking into account the latency in the visual system. So that's kind of what I want to present here about thinking about what's happening when the world moves. So we've taken kind of like a reduced form, just saying, okay, we're interested in these sort of more complex aspects of vision, but we're still going to keep some control. We want to understand how we can compare whatever we're learning from static to whatever we're learning from dynamic. And this question of generalizing from static to dynamic. So what I've shown you is that we're getting better generalization from the later part of the static response. And what I want to argue is I think that this early part of the visual response to flash stimuli may be more about flashing than it is about really the real properties of the stimulus that are relevant to natural viewing. And I think we're introducing this very strong sort of transient into the responses that may be misleading when we're thinking about continuous kind of visual processing. I also showed you here that in the course of a movie, the errors kind of accumulating over time. We're falling further and further behind in terms of relating the static response to the dynamic response. This may be reflecting kind of issues that, when I talked earlier about the study from David Leopold's lab, they're interested in sort of temporal continuity, right? Again, these later orientations that we're seeing are preceded by lots of other orientations. There's lots of things that may be going on here. Things like adaptation and stuff like that are also kind of relevant. The predictability of the stimulus, these are all kind of things that are relevant. We can't really dig into those here, but I think it tells us this is stuff that we really should try and understand if we want to care about natural vision. And what appears to be an initially somewhat predictive response, right? It's slightly predictive because of these lags in the visual processing, starts to lag over time. And the other thing I want to suggest here is one thing that I think is becoming increasingly common, more and more people are going to sort of dynamic visual presentations. And oftentimes now we see people using sort of short video clips as a way of presenting dynamic stimuli, right? And that's what we're doing here. And for MEG, we can actually look at how the response is varying over time. But I think short video clips in general may overemphasize this onset response. And I think this is particularly relevant for fMRI because in fMRI, if you present you short movie clips, I think what you may be getting is still that large transient response to the onset of the stimulus. It may not be so much reflecting the dynamic aspects of the stimulus and what's going on there. So as I say, this is sort of us sort of dipping into this, going into the dynamic world with a lot of trepidation, trying to maintain some control and actually trying to get our heads around this bit is realize the complexity of thinking about what's happening in the sort of dynamic world when you've got stimuli moving. So this is for us is kind of a first step, but I think it highlights some really important issues that we need to think about. So that's what I want to say about the world moving. Okay. So now, yes, so thinking about natural vision, I said, well, okay, one aspect is the world moves. Second aspect is we move. So again, this is another level of kind of complexity in terms of thinking about this. And so we move our head, we move our body, we move our eyes. And the work I'm going to present here is work that was led by Grace Edwards, who's a staff scientist in my lab. And this is in collaboration with Eli Merriam, who's another investigator here at NIMH. So, and we were particularly interested in saccades. And saccades are critical for vision, right? We move our eyes in a very directed way. So saccades allow us to efficiently sample the visual world. We move our eyes around to pick up different parts of different information. We can select the features of the visual world that we want for inspection. And one critical thing here that I think relates to thinking about sort of the classic paradigm for object vision is that actually when we foveate something, we already have information about that foveated thing. We don't look at something to be like, oh, what's there? Right? We move our eyes towards something that may be of interest given our current goals or given kind of, whatever else is going on. Maybe there'd be some bottom-up reason why that would be informative. But we have information about a to be foveated feature before it actually lands on the fovea. And that's very different from the situation where we're flashing up stimuli that has no prior context. So nothing is really flashed on the fovea, right? Again, moving your eyes to a point in visual space is not the same as a stimulus suddenly flashing up on the screen. Now, one thing we know, because we know a lot about kind of visual cortex, this is sort of to make this kind of general point about what it means that we're moving our eyes. So this is like a demonstration from Jonathan Horton and colleagues. And so here, what I'm showing at the bottom here, this is sort of flattened representations of the left and right visual cortex. The foveal representation would be here and here. And this is like a stimulus that someone may be looking at. And this is like the fixations where someone is looking from the red cross. And we can show how that stimulus is mapping to the cortex. And one thing to note here, because everybody here is a vision science geek, is that here things have been flipped, so it's not going to be inverted. But just to give you an idea of how what's going on. So in this clip here, as the person is moving their eyes around, we know that where the information, the visual stimulus is being processed in the visual cortex is changing moment to moment. And of course, this is a real challenge for thinking about how our sort of natural continuous visual processing occurs. So to sort of talk about this in a slightly different context. So let's imagine that you're driving down this road here. And when you're driving, some information is particularly relevant. So in this particular case here, this may be really important for your current goal, right? We need to keep in mind what this light is showing, whether it's green, whether we can still go, whether it changes to red. But at the same time, we're sort of attending to this, but we're also looking around at other parts of the visual world. And so let's imagine that now we're going to make an eye movement here, but we know that this light is kind of important. And so there's a kind of, we may activate a receptive field that corresponds to where the traffic light is going to land in the visual field. And this kind of process here is something that people have talked about in terms of saccadic remapping. And in general, there's evidence out there suggests that the brain maintains the spatial information across saccades. So we're not just making these random things, there's some maintenance of that spatial information. But actually, it may not just be spatial information, because actually we may want to maintain those features. So it's not the case that this light is just a spatially relevant location. Actually, the nature of this is also very important. So we're actually kind of maybe as we're moving our eyes around, we're still maintaining some attention on this particular traffic light. And so when we're moving our eyes, it may be important to also not just keep in mind that spatial location, but the feature information, so that when we have the receptive field that's going to land on the traffic light, information about what is going to land there when the saccade ends may also be transferred. And there's also some information out there that suggests that the brain does maintain feature information across saccades. And I'm not going to discuss that kind of here, but sort of focus on kind of the question that we had in this study, which is what we were interested to say is, okay, can we find evidence for this kind of feature maintenance across saccades in primary visual cortex in V1? Are we seeing this influence in the early parts of visual cortex? So to take you through kind of our study. So participants in this study, this was an fMRI study, and participants are fixating on the right side of the visual display. An image is then flashed up and appears in the periphery. In this case, it's going to be in the left visual field. The fixation cross then moves left across the screen, and that fixation cross moving cues the participants to make a saccade. They saccade now to the left side of the screen. And of course, when they do that, the visual stimulus is now going from the left visual field to the right visual field. And the participant's task is to say, okay, does the stimulus stay the same or not? So is the stimulus that was present before the saccade, is it the same as the one that's after the saccade? And so what's actually happening during these trials is that contingent on the saccade, we will either keep the stimulus the same. So during the saccade, the stimulus actually will briefly flash off the screen and flash up again, but the participants may not be aware of that, but the stimulus remains the same. On some trials, we can change the stimulus. So in this particular case, there's a sort of a very simple kind of stimulus setup. The stimuli were actually either a face or a place. And so we're going to switch either from the face to the place or the place to the face. So we have these change trials so that now after the saccade, the stimulus that's present is different. Or we can just disappear the stimulus during the saccade. So now when the eyes land on the left side of the screen, there's actually no stimulus in the right visual field. So these are the different conditions that we have. And just to sort of give you an idea of like, what are we looking at in here? What are our hypotheses about what's going on? So here now I'm showing you the brain here. We got the right hemisphere here, the left hemisphere here. This is our setup here. So initially, people are fixating here, which means that we have no stimulus in the right visual field. So nothing in the left hemisphere. We have the stimulus in the left visual field, so processed by the right early visual cortex and right V1. So now we have the saccade, and the stimulus is now effectively moving from one hemisphere to the other, because now the stimulus is in the right visual field, and there's nothing in the left visual field. So this is what's happening. So on our same trials, we're keeping the stimulus the same, pre and post saccade. So what's in the right visual field matches the expectation of what would be there pre-saccade. And if we now apply kind of a decoding style analysis saying, based on the pattern of response in V1, can we decode whether it was a face or a scene or a building? On these same trials, we think that we should be able to decode what's going on. So we're looking in left visual cortex and trying to decode what's present. So now what about the change trials? So on the change trials, again, pre-saccade, the stimulus is in the left visual field. Post-saccade, the stimulus is now in the right visual field, but it doesn't match the stimulus that was present pre-saccade. So if there is transfer from pre-saccade to post-saccade, that might interfere with processing and our ability to decode what is present after the saccade. And so here we get this mismatch between the pre and post-saccadic stimulus. If there is transfer, this should impair our ability to decode what stimulus is present. Finally, we can say, well, what about the disappear trials? So in the disappear trials, so you've seen this before, we get the same thing going on here. Pre-saccade, the stimulus is in the left visual field, not in the right visual field. So being processed in the right hemisphere. Participants make the saccade. There's no stimulus post-saccade now. But if there is transfer, we might expect that actually some information will actually now be present in the left visual cortex, even though there's no stimulus in that right visual field. And so here we might predict that, okay, maybe in fact in these disappear trials, we would also, if there is this transfer information, we would also see some ability to decode what stimulus is present post-saccade from the left hemisphere. So I hope this is clear. I know this is kind of a little complicated, but I hope this sort of makes sense given these predictions. Now, in this kind of study, right, the critical thing here is we're in the MRI and we're having people make saccades, we're trying to do eye tracking, and we're presenting stimuli contingent on where the eyes are. So it's actually really critical that we only have trials and only have participants who are actually following our instructions. And so just to give you an idea of this, this is an example here. So on the left here, this is an example of a good participant. So the vertical lines here are corresponding to the time of the saccade. So we want people to make a saccade at this time. And you can see this person makes the saccade and holds which is good. We got 19 people who can do that. This is an example of a bad participant, somebody who's not really able to follow the instructions, or in fact, our ability to eye track is not as good, where we don't have evidence that they're making the saccade at the right time, so we had to exclude these. And actually, this experiment was a real heroic effort from Grace to go through and run many more people that we could actually include in the study, because this was so important to the design to have this. And actually, we included participants who only achieved a certain proportion of trials. We also got rid of trials where we had evidence that they hadn't made the saccade at the time we wanted them to, relevant to the stimulus. Now, to look at the data, what was kind of important was that we also want to say, okay, where in V1 are we expecting to see the response? Now, the visual stimuli, what I showed you before is the visual stimuli always appeared in the upper visual field, but actually we presented stimuli both in the upper visual field and in the lower visual field on different trials. And so we had simple localizers where we present just a flashing checkerboard, either in the upper visual field or in the lower visual field. And it's the right visual field because we're going to be focusing on the left hemisphere. When we do that, we can identify these ROIs that correspond to the stimulus location that's relevant on the individual trials. And so on a trial where the stimulus is in the upper visual field, that upper ROI is sort of our target ROI. And this lower ROI is when we can ask, okay, how specific is any information that's transferred? Is it specific to the location that's relevant, or actually does it generalize throughout the visual field? So we can compare this and look at the specificity of any signals that we see. And when the stimulus was in the lower visual field, these were obviously flipped. Okay, so now we can look at the data that we got. So I'm going to show you the decoding accuracy for the same, the change, and the disappear trials. And chance here, because we're just decoding the difference between a face and a building, is 50%. So here, this is what we saw on the same trials. So in this particular case, we actually are able to decode, which is what we expect to be able to do. This again, people are making the saccade. There's a match between the stimulus that was pre-saccade and post-saccade. So this is good. We can decode in that kind of condition. But what about the change trials? These are the critical ones where now we get this mismatch where if there is transfer, that should interfere with our ability to decode the stimulus. So on change trials, we've lost our ability to decode what stimulus is present. We can see here for the individual participants, most participants showed the same drop from same to change, and this is statistically significant. And what about the disappear condition? So in the disappear condition, you might think, okay, if there is transfer, maybe we can pick it up in that disappear case. The results we get here, it's not significant. It's somewhat ambiguous. We can't say too much there, but it appears that when there is no stimulus present afterwards, actually we don't see clear evidence to be able to decode in that particular case. Now, I told you before that actually the targets or the stimuli could either be in the upper or the lower visual field. These data are actually collapsed across those two. But we can break it down and say, okay, in terms of spatial specificity of the signals that we see, is it specific to the location where the stimulus is, or does it actually generalize throughout the visual field? What we actually see here is so information transfers across the saccade, and actually it looks the same in both those ROIs. So what we're seeing is actually a spatially non-specific signal in this particular case. So it may be that whatever is transferred is not about that very specific particular location, but is some feature that is transferred across the saccade. So this is great. We can do this, but do we even need a saccade? What if we run the whole experiment, we have no saccade, just to make sure that actually it is relevant to that? If we have no saccade, then we don't have any decoding. So we know that a saccade is needed. And so what I've shown you here is that we now have some evidence for feature maintenance in V1 across the saccade. So there appears to be this pre-saccadic information transferring and impacting post-saccadic processing. When the stimulus disappears, we don't see strong evidence for that transsaccadic signal in this particular case. And we have no evidence for that information present in left V1 without the eye movement. So just to sort of quickly summarize what I've shown you here. So in terms of thinking about object vision in a dynamic world, I've shown you like a focus on thinking about the world moving and thinking about how we move. So when the world is moving, sort of raised this issue that actually we've got to be really careful in thinking about how what we've learned from static visual processing with flash stimuli may generalize to sort of natural vision. And on top of that, because we move our eyes in natural vision, we also need to think about the fact that every new fixation isn't like a different snapshot. There may be this information transfer even in somewhere like V1 that's happening. That's also relevant to thinking about how processing occurs sequentially in kind of natural visual situations. And what I really addressed here is this question. And I think, I mean, I've given some insight, I hope, into how we think about this, but I think this is a big open question. What is it that we've learned about static visual processing that really generalizes to natural viewing? How much can we take from that to think about what's happening during natural vision? So I'm going to end there. I just want to thank, this is actually just the people over roughly the last 5 years in my lab. The people who are highlighted in red are the people who are currently in the lab. And without them, we couldn't do any of this work. And in particular, the work that I presented here was done by Shruti Chippy, Rohini Kumar, Grace Edwards, and then Eli Merriam collaborated with us on that final study. And thank you, and I'm happy to take any questions about all of this.
Thank you so much, Chris. This was really wonderful, insightful, and I think a really important step in assisting us in moving to a more really comprehensive understanding of how vision is in the real world. And so, first of all, I want to invite everybody to unmute yourselves and let's give Chris a big applaud. And also, yeah, we're opening. So, Jeremy, is that your hand raised?
No, it's actually Zoom's hand raised. This is my
Okay.
hand. It's different. You can tell because this hand is dynamic,
I know.
which Chris can decode while it's.
Okay,
So
great.
were you asking whether I wanted to ask a question? Was that the actual purpose here?
Okay. Were you?
So
No,
yes,
'cause,
yes. So my question for these last experiments is whether or not you need to be paying attention to the stimulus on one side of the saccade and the other. So I'm sort of imagining,
Yep.
Suppose you had a vertical line of objects, and
Right.
you can let them freely attend or something like that, and then can you still decode just any old stuff that's moving from the left hemisphere to the right hemisphere, or does it have to be task dependent?
Yeah. So I think, and this is a great question. And, to some extent, we're cheating, right? Because we have a stimulus that flashes on the screen. right? So we were capturing attention, presumably, when that comes up. When people are fixating, that stimulus is flashed before they make the saccade. So, you know, in the example, when I talked about the driving, there I said it's like it probably is relevant what you're attending to, right? Do we think that everything is kind of, if everything is moving around, this is an incredibly complex situation. So I think it's a really important question to say whether it is only what you're attending to or other things. I can't answer that with this experiment here, but I think it's actually really important to think about that situation. And I would guess that it should be prioritized towards what you're attending to, but we need to run studies like that to find out.
So the follow-up to that is that you were talking about features, but you were using faces and places, which in my world might not be considered to be
Right.
features. I wonder whether you would get more evidence of preservation of sort of front end features like color or orientation
Right.
across the saccade without attention, whereas knowing whether a face had turned into a place or whatever, if you controlled for those front end features, might in fact require attention.
Right. I mean, potentially so. I mean, I think part of the reason to go with something like faces and scenes buildings here, right, was because actually we sort of actually tried to minimize the stimuli are not particularly differentiable within V1 as we created them, because actually they were kind of matched for spatial frequency. We kind of sort of
Yeah,
controlled them
yeah.
in different kind of ways. Because we actually wanted to sort of see the case where information really has to move around, right? It'd be very hard for it to go from V1 to V1. So we think that actually what we might have here is much more of a higher level signal being transferred. But again, we haven't tested all these different kind of things, right? I think these are really kind of critical questions that I can't really address from this point. And I think, again, the nature of the kind of information that's present in the stimulus and how that relates to the particular visual area will also be particularly relevant. But I can't address that with this study.
Were you also looking in face and place areas?
Yeah, I also wanted to ask that.
We do. I mean, the it's sort of like we have in those cases, so we have like some evidence that we get the differentiable signal in those kind of areas. It's quite weak because this is actually a very, you know, because the stimulus is only present very, very briefly, and because in fact it's actually quite low contrast and so on. So we can do it there. But again, we sort of realized after the fact that actually to be able to really address those questions, the stimulus setup that we had is not ideal for that. I mean, the challenge that we've had here is this study I said that it was heroic. This took several years to actually do this study to get the people through. And so we're trying to think about now to test these more refined questions. We're sort of thinking about different kinds of approaches, right? So actually here, one thing we're thinking about doing is moving away from MRI to trying to do this with something like EEG or MEG, which may actually give us a bit more control, but actually the way in which we have to do the experiment is now going to have to vary. So we'd like to go in and look at these questions in other ways. But in this study itself, we can't address those things.
Yeah, it's always the nice part about asking questions. You can ask, fairly simple sounding questions that
Yeah,
the speaker knows would require the next 5 years of his life.
we'd love to do them.
Um, Chris, I also want to, oh, I see Asaf has raised his hand, so just a sec. I want to ask, following up on Jeremy's question about the issue of where, I mean, I understand you probably did not localize FFA and PPA. I mean, you showed just the localization with checkerboards, but I'm wondering if you do use even anatomical coordinates or use, all of the visual cortex or look into,
Yeah,
additional retinotopic regions or perhaps even do some kind of like, all brain analysis, seeing if you can decode it from anywhere. I don't know,
yeah.
maybe even. So would you find somewhere
So
that you would be
so
able to decode it from?
we do in those cases. It's not strong, but we do find the evidence when you try and use those. Again, we tried to use the ROIs from, again, they're not ideal. We can try and do like a
Yeah.
search kind of thing. So we do get some evidence that we get those peaks in those kind of areas. So I mean, that's just a general sense that this is actually the information that's been transferred may be going through, higher order areas and becoming top down, as opposed to thinking about something that's just going, you know, somehow just because it's simply closely.
Yeah, great. And okay. So Asaf has I have another question, but Asaf, are you here? I mean,
Yes, I'm here,
okay,
and
good.
I apologize for the lack of camera. So great work, really interesting. But coming from a more, theoretical kind of perspective, And it's not really ordered, but still. So why actually separate the actor or the observer from the world? Because basically, I mean, if you think about, ecological psychology, the whole idea is that as you move about the world, the actual information, right? I mean, is changing as well. So it's basically a dynamic change, and that's going to be accompanied by whatever feedback loops and whatnot. So I guess my question is, isn't the separating this dynamic vision as you call it into two parts actually is still rather artificial? And then relatedly, just because of, this kind of, I'd say bunch of stages of generalization, isn't, for example, decoding from static object to dynamic object is still limited in the sense then you'd have to ask whether a dynamic object generalizes to, dynamic scenes and whether dynamic scenes generalize to the world. So isn't it just another, maybe a bit wider than a snapshot, but still a bit of a snapshot view of natural vision? Sorry if
No,
it's too
no,
philosophical.
no, that's totally fine. I mean, no, all of this is true. I mean, I completely agree with you, right? On all these points, right? I mean, there are so many factors that are actually that are relevant to natural vision, and I think that what we're trying to do here, and there's different approaches that people are taking in the field, right? I mean, there is this general recognition that actually we do want to think and sort of more take on the complexity of what's out in the world. It's a question of how you do it, and what we're trying to do is find a way where we sort of think about what's relevant but now hone in and ask more targeted questions about that. It means we do come down to these slightly more limited kind of experimental setups. And in part, that's because I think that if we jump into, and there are people out there collecting data from human participants with intracranial recordings moving around in the world, I think that actually it's just incredibly complex to break that down. So we're trying to say, okay, there's lots of things that are relevant, but we want to try and focus on these particular factors to help get some traction that helps us move to the next step. But it doesn't end here. And I think your point about, yes, there's all these different things. As we're moving through the world, there's so many other things that are going on that actually we need to think about. And I think, again, the issue of going from the static to the dynamic is really not because I think that's relevant when we want to ask, given what we think we know about vision, if all of that was only about the static context, is it relevant for those other situations? That's partly why we're doing this. I think in the end, no, the static situation, because it isn't like sort of the way in which we actually experience visual input typically is incredibly limited. But we do need to understand how what we think we know now is relevant or not for the dynamic world. So the conclusion that David Leopold's lab have come to, where they basically say, look, there's a completely different mode of operation that's happening in natural viewing. That would be a real challenge for us because it says, okay, well then what do we understand? If it's a completely different mode and static viewing isn't like normal viewing, where actually are we? So I think it's all about, I think the philosophical questions are really important, but actually in terms of trying to get traction within this, we still kind of need to break things down. And that's kind of the approach that we're taking. So trying to think, what are the relevant questions, but how can we do that in a way that allows us to gain understanding in a more controlled situation?
No, I understand. I guess what I'm wondering, and I'm just again thinking out loud, is whether it really advances our understanding of natural vision, or it advances our understanding of the limitations of our experimental paradigms.
I hope there's a bit of both.
Okay. I
But I
hear
think
you.
there's a lot more to go in terms of understanding natural vision, right?
Yeah.
A lot further we have to go. I say to my mind, I mean, we've been hesitant about going down some of these angles because we sort of understood some of the complexity. But now that we start doing it, you start to realize all the questions that arise as you do this that makes you really wonder, like, okay, do we need to do things in very different ways? But again, it's challenging, right? Going to a completely uncontrolled situation raises all sorts of other problems as well.
Great. Thank you.
Yeah, the more we know, the more questions we have.
Right.
Are you happy to take Galit's questions? I mean,
Sure.
yeah. So Galit.
All right. Yeah, just a quick one. So you think that the problem for the prediction from static to dynamic is not the static itself, it's just the onset? I mean, so once you just ignore the onset, right? And you present the
Right.
stimuli for 5 seconds, 10 seconds. Because at the end, in the real world, we do look at photos, right? At least in modern vision, right?
Right.
So it's not that unnatural. So the problem is just the onset, right? I mean, the onsets are unnatural, but static, could be natural, and you can learn from it about dynamic, right?
Yeah. Yeah, no, I think so. I mean, I really do think that the onset is a real challenge for us, right? I think that's a real problem. I mean, in
Hmm.
terms of like if you're studying a photograph or whatever, right, you're moving your eyes though, right? So having fixation at a prolonged presentation is, I don't
Right.
think, natural.
That's not
But
the
that's why,
same as
again, I think
onsets.
this question of then thinking about eye movements, and again,
Hmm.
that's something we're thinking a lot more about is like how to introduce eye movements into the kind of paradigms that we do in a way that still allows us to sort of understand what's going on. But
Right.
yeah, I think thinking about all of this and doing this kind of work has made me really wonder about about the onset. And I think because in general, whether we're doing, MEG, EEG, whether it's neurophysiological recordings, right?
We have to do
The
onsets.
dominant part of that response is always this first peak, and that's what we always tend to focus on because it's the strongest part. But if that is really about onsets
Right.
and not about like really ongoing processing in the brain, then actually I think that does raise a question for how we want to think about it, things.
Yeah, totally agree. Hmm. Thanks.
Great. I mean, yeah, I also have a question you said, which I wasn't aware of, about the fact that the stimuli, the face and house, were actually equated for spatial frequencies. First of all, that's amazing. To equate faces and houses. But do you think that the results, because V1 is so sensitive to high spatial, or anyway, sorry, to spatial frequency distribution, then this is why I mean, if you would have used just, let's say, naturalistic stimuli, do you think that the decoding would have been higher? I mean, like you've gone against yourself in a way, like
Exactly. And I think it's one of those things that we designed it this way and everything later on realized that this has made it very challenging for us to actually get the result right. It really now has to be something that probably could not be generated easily within sort of the
naturalistic
population as well.
inputs are, yeah, are
So,
much
so
more
in that case,
distinct,
there probably would be much more information that's present.
even in V1.
Yeah.
Yeah.
But again, we'd have to look at that. And I think it's still the if we do that and we found a slightly stronger decoding, I think the result would still hold. Again, it's one of these things where you go back and you think like, well, why did we design it this way, 3, 4 years ago? And yeah,
Yeah. Well,
and there's a reason, right? But it's yeah. So I think it's an interesting question to think like, okay, what would we see in that case if we now didn't sort of introduce all this sort of artificial control of the stimuli?
but by the way, I have another question. With respect to, you said you had top and bottom stimuli, and you showed that the accuracy was kind of like the same, even if you divided divided the classification between upper and lower, specifically for that. But what about if you examine the temporal aspects, like because I don't know which. I mean, I'm wondering if there are differences in the temporal, let's say, envelope or the temporal peak. If you look at that, I mean, I know the
Yeah,
signal is very sluggish, but
right. Yeah.
so
No,
maybe there's an. But I'm
I think
just
we
one.
can't. I think in this one with fMRI, right, it's really challenging to kind of look at that. And again, I mean, what we can say here is that it's not specific to the particular location of the stimulus. But of course, both of those locations were relevant in the experiment. Is it a global signal? I don't know. We again, because with the way we wanted to do, we want to focus in on those particular regions where the stimulus is. So, but maybe it's a global signal, maybe not. I mean, we can't fully answer that. But I think this issue of looking at like the temporal aspect is kind of key, which is why instead of moving forward with this, we are thinking about how can we do things using something with a much faster um, temporal measurement.
Thank you. This is really interesting, and I think it's a really important phase that the field has to go through from going all the way. Like these are steps we have to take in order to understand. If you're looking at the Leopold's group study, like where is this? Where is this gap? And so I think this is really exciting. These are exciting findings and a really interesting really interesting work. So thank you so much for joining us
Thanks very
and
much.
presenting your work. It's really fascinating. And again, thank you for joining us. And next week we will have Sarit, which I think is here, muted and unseen. Sarit Shapiro from Haifa University. Yeah, hello. She will be talking about vision, low vision, would we say? Would low vision and assisting low vision and new techniques. And so again, see you all, well, not everybody, next week. And thank you so much, Chris, for joining us. And have a
Okay.
nice rest of the week.
Thanks very much for the invitation.
Thank you.
Bye.
It's been a
Thank you,
pleasure
Chris.
having you.
Bye.
Thank you so much.
Bye-bye.